copies of target DNA, you will choose a vector, right? What it should have? If you want to recover many copies of target DNA, you will choose a vector which does not have an origin of replication. It wants, then only replication will start. Which has antibiotic resistant gene? Okay. Whose origin supports high copy number? Yes. Which has only one restriction site? Yes, it is your C. Actually, if you want to recover many copies of target DNA, you will choose a vector. Vector should have an antibiotic resistant gene also. It should be able to produce a high copy number also, and it has only one restriction site also. Actually, all are right. The question is wrong. Question is wrong. Next, 14th one you go in. Choose a correct pair for the following. Polymerase breaks the DNA into fragments. What is the function of polymerase? What is the function of polymerase? It adds the nucleotides to it. Okay, so that is wrong. Next, nucleases separates the two strands of DNA. No, then what does nucleases does? Nucleases, aces cleave, nucleic acid, DNA or RNA. They are saying nucleases separate two strands of DNA. Right only, no? Yes or no? Say yes or no. No. Actually, it is no, because nucleases specifically cleave phosphodiester bond. Remember that. So that way it is wrong. Got it? Next, exonucleases makes cut at a specific position within DNA. No, at the end. Ligases. So that is your D. Okay, but nucleases, remember, phosphodiester bond, it will cut it. Next, which of the following tool of recombinant DNA technology is incorrectly paired with its use? EcoR1 produces sticky ends? Yes or no? Yes. DNA ligase multiplication of rDNA molecule? No. DNA ligase multiplication, no, it is joining. Then DNA polymerase and polymerase chain reaction amplifies? Yes. Selectable markers? Yes. So the answer is B. Now, what are the two antibiotic resistant genes located on vector? Okay. Then next, match the column now. 17. Whatever you know, see the hint is that whenever they give 4 options, please remember 2 will be your short you know that it is wrong. Your confusion will be next to 2. Out of 4 options, 2 surely you can say that it is wrong. The other 2 optionally, 1 will be a distractor. It will distract you. It will give a wrong, the, a, non, hyphen, like that, they would have not put a comma, like that they will confuse you to select the option. So if whatever you know, if they give in a matching, first to do that, automatically the unknown you will come to know. Got it? Select now. Tag polymerase. Stable about 90 degree. Exonucleases cleaves the ends of linear DNA. Proteases, protein. Chitinase. So the fourth one is the option D. It's not there. I mean here in the column B. So it is your option D. Okay. In recombinant DNA technology, the term vector refers to. What is the term vector is referring to? It's a plasmid that is used to transfer the DNA into a living cell. Got it now? Next. Which one of the following characteristics is generally not preferred for a cloning vector? Multiple. So what are the characters it should have? Origin, high copy number, selectable markers. Okay, these 3 should be there. Next, the following statement describes the characteristics of an enzyme restriction endonucleases. Identify the incorrect. Identify the incorrect. What is wrong in A? Who told A? What is wrong in A? What is the answer for that? C? palindrome nucleotide sequence. Correct no? The enzyme cuts DNA molecule at a different identified position. Correct only no. That is what restriction Indian nucleus also does. Correct only no. That also is right. So what is the incorrect one? Already we have done C. Already we have done B. Then A, B is left out. Now tell A or B. Actually the answer is A. Right. Whoever has told first. But when I asked what is wrong in that, it has given it cuts only one strand. No, both the strand it will cut. That is the answer. Got it? So read. A is the incorrect one. It cuts only one of the two strands along. It cuts both the strands. Next, who among the following was awarded the Nobel PCR? PCR. Caribulus. Next, during the process of gene amplification using PCR, if a very high temperature, read and tell me the answer. That's why I stopped over there. Tell me. Denaturation. Actually, when you read no, not you will miss her, which is not maintained in the beginning. Then denaturation won't happen. They don't get sweet. Wake her up. Next. While isolation DNA from bacteria, which of the following enzyme is not used? Why? Why it is not used? Why it is not used? When you should have an answer now when you say. Why it is not used? It will cut DNA only. So why you will use it? You are isolating a DNA. If you use an enzyme, it will cut a DNA. Why you want that enzyme? Next. Which of the following steps are catalyzed by Taq polymerase? Which step? A, B, C or B? All of this. All of this. It is C. Denaturation, we want to separate them. Annealing, we will add the primer to it by reducing the temperature. And then extinction is by adding the nucleotide. Taq polymerase. You have studied DNA polymerase in your molecular biology. In the molecular inheritance we have studied, right? We have studied in replication also, we have studied it, right? DNA polymerase, it polymerizes by adding your nucleotide. Same thing. Only difference is Taq polymerase can withstand high temperature. Next, which of the following is not an application of PCR? Purification of isolated protein. Correct. Plasmid used to construct the first recombinant DNA was isolated from? That is what I told. Read this question and answer. I told. Plasmid used to construct the first recombinant DNA. Plasmid. E. coli. That is what I told. Gene was from Salmonella, but vector was from E. coli and inserted into E. coli. Host is also E. coli. Understood? Next. Name the drug used in cancer treatment produced by Biotechnology is? Cancer treatment. Insulin is diabetic. What is TSH? What is TSH? Class 11th. Endocrine system. What is it? Thyroid stimulating hormone. TSH. Yes, sir. It is interferon. It is interferon. Okay, actually to say insulin, interferon, all are produced by biotechnology, but which is used for cancer treatment. So it is interferon. What are interferons actually? Antiviral protein. Why we are using for cancer? They are antiviral agents, correct? Got it? What is the property of a virus? Now we are deviating. They are obligate intercellular parasite. They want a host. Already virus attack is there. Protect yourself. That is why it is responsible for your own immunity, plus it is antiviral and we say because following the viral infection it has been produced. Next, the requisites for biotechnological production of antibiotic is, what are the prerequisites or the pre-requirements? All of them. Is it true? Is it all of them? You have to search because you want it. Then next, isolate an antibiotic gene. All of these actually. All of these. See, whenever they ask is it right, you will have an answer based on which you have selected it, right? You should be able to confidently tell that. Then only you can tick in the exam. Otherwise, you will also think twice, you will have an option. Humulin is prepared by which type of option? B. Clostridium. Did I write Clostridium? I wrote, see, E. coli A, E. coli B. But he is very smart. He has given entamoeba coli. What is entamoeba coli? It will cause amoeboid descent. It's a protozoan, but he will not cause. It is entamoeba histolytica, but same protozoans. Okay. Next, during the purification process of recombinant DNA technology, addition of chilled ethanol precipitate out. There should be no doubt in it because it's a practical. We have done it. We have seen DNA, yes or no? Don't say no. Yes, no. It's a practical, right? We add ethanol, DNA will precipitate out and come. Got it? We add chilled ethanol. And they may ask sometimes, why chilled ethanol? Why chilled ethanol? If you don't add the chilled ethanol, the stability of the DNA will be lost to stabilize the DNA. Got it? What are histones then? Anyway, it is DNA technology, RNA ruled out. Polysaccharide also ruled out. So between DNA and histones, what are histones? Positively charged proteins. Right, made out of what they are made out of? Why they are positively charged?
Because of lysine and that.
Arginine, remember lab. Okay, lysine and arginine, short form. Got it? Lab you can remember. Next, so the answer is D. DNA is negatively charged, histones are positively charged. For compact packing of that you require them. But here it is DNA. Which of the following is not related with bioweapon? E. coli. What about Yersinia pestis? What does it causes? What does Yersinia pestis causes? Disease, madam. Disease it is. Yersinia pestis. What does it cause? Plague. Got it? Plague it will cause. It is a spread. It will spread. Okay, Bacillus anthracis, it's a biological warfare. Clostridium is also, because whichever organism is a spore former, you can use them. So it is E. coli. E. coli is the not related. Okay, out of these three, which one you will rank first? If it is a bioweapon. If it is a bioweapon, not bioweapon. If it is a bioweapon, which one you will rank first? Anthracis. Bacillus anthracis you should name first. Next, transgenic animal as? Foreign DNA in all its cells. So first now, how does Agrobacterium tumefaciens will insert the DNA? That is the how does? So insert a gene into the plant, we are using Agrobacterium. Yes. Now, how Agrobacterium tumefaciens is inserting? Tell me now. You know the vector. T and plasmid is there. It is inserting. But how it is doing it? You should know it. No idea? First now, Agrobacterium tumefaciens is a bacteria or a, what is it? It's a, Agrobacterium tumefaciens is a bacteria actually. Shh, listen now. For plant, the vector is Agrobacterium tumefaciens. It is a bacteria. The bacteria has a plasmid also. Plasmid is? One of the speciality of Agrobacterium is that naturally it can infect a disease in a plant. Say this is your plant. Naturally it can infect certain plants and it will cause root gall formation. Usually at the lower part of the plant. So wherever it has inserted in, that cells will start to multiply, so you will have a gall formation. This is a natural process. You are able to get it. Now what we will do is that I will take this Ti plasmid out. Insert my gene of interest. Got it? Again I will put it back, place it back into Agrobacterium tumefaciens only. Now it has my gene of interest. Let it infect the plant. Whichever cell it infects the plant, that cell will not have my gene of interest? That's how they will do it. Correct? But it will not cause gall formation throughout the plant. It will cause only in a particular cell now. But what do you mean by BT cotton? That means the whole plant will have the gene which codes for the cry. But here only a part will be having it. So after inserting in this gall formation, they will take few cells from it. Then they will go for micropropagation. What is micropropagation? Huh? Yes, growing a plant by tissue culture. So that one cell which has my gene of interest, it will multiply. It will have a callus formation. Got it? Then from that my whole plant, the one cell is going to multiply, it's going to be a callus. Then I am going to put it back into a rooting and a shooting media again. So the whole plant is going to develop because the initial first cell had my gene of interest. The whole plant is now going to have my gene of interest. That's how they do in case of Agrobacterium. Remember, for Agrobacterium tumefaciens, plasmid is also from it. Host is also your plant. But again, placing it back into your Agrobacterium tumefaciens and then natural infection. That is a boon. Naturally they can infect. Till now. Next question, 33rd question. Which of the following is a correct sequence of steps of PCR? A. Next, E. coli used in the production of interferon. Then what is that LH? What is that option LH mean? Luteinizing hormone. Then what is that egg Dyson? No idea. Okay, what is that egg Dyson? Actually, you should add E to it. N-E you add. Typographical error. E you add. Now you see whether you know. It is a molting hormone for insects. Okay, molting hormones for insects. So it's not at all for humans. So obviously it is your interferon. Next, the bacteria generally used for genetic engineering is Agrobacterium. Genetically manipulated organism containing in its genome one or more inserted gene of another species is a genetically manipulated organism containing in its gene one or more inserted gene of other species. The word that you have to see is not just a gene of interest. It is one or more inserted gene. More than one gene is inserted here. So it is transposomes. Got it? Transposomes, jumping genes. They will leave that place, leave their gene, and again into another gene also, into another place also they can jump in. Then what are retroposomes? Retroposomes, what are they? They are variant of transposomes. Remember, they are also transposomes but a variant of them. Next, in a mixture, DNA fragments are separated by? Yes, right. Production of human insulin is from? Gut bacteria. Actually, they have not given E. coli. Got it? Which one we call them as a superbug? Which one is a superbug? Which bacteria is called a superbug? Pseudomonas. Pseudomonas, if they ask. In bioremediation, you will study this. Another chapter where you have the human health and welfare, human welfare. There, sewage treatment you will be studying in, right? So there you will be coming across with this. Bioremediationally, you will come across with superbug. Even they will ask which one is called as Chakraborty's bacteria. Pseudomonas only. Okay, because he first isolated it. Okay, now find the incorrect match. Find the incorrect match. TPA, tissue plasminogen activator, erythropoietin, formation of RBC, interferon treatment of cancer, Riopro transplantation rejection. Tell now, which one is the incorrect match? C. What made you to think it is C? Because other three is right, we know. So left out is C, right? But actually to say, Riopro is a recombinant protein which can detect acute kidney transplantation rejection. It's a recombinant protein which, when a kidney transplant is done, when your body is not accepting that kidney, that rejection can be detected by using this protein. So it is a right option. Now search for the wrong option. C is right. Huh? A is wrong. Why A is wrong? Because when I am giving explanation. You should also give. Why A is wrong? See, it is blood clot preventer, but it is blood clot buster. There is a difference. Buster means clot is formed and bursting. Preventer is not allowing it to form. That is a word. That's a wrong. So it is 39A. 40? 40. Read the question properly now and then. Read the first question. I told hepatitis is a recombinant. Now you are telling what is the question now here? Which is not made from? Tell now. Anthrax. C. Okay. Foot and mouth disease. That is for cattle, not for humans. Foot and mouth disease for cattle. Vaccine is a recombinant vaccine. BCG is for? BCG is for? That vaccine is given for? Tuberculosis. This one. What is that? Mycobacterium against Mycobacterium tuberculosis. TB bacilli. Against TB. BCG. BCG is the first vaccine that has been given when a child comes out. Immediately as soon as you come, first vaccine that they give. What does BCG stands for? Bacillus cholesterol. Bacillus calmette guanidine. Okay, Bacillus calmette guanidine. Okay. So it is anthrax. Next, ethanol is produced from starch as a raw material by the activity of yeast. Super. Maximum utilization of the technique of biotechnology we have seen in which field? Is it? It is more in agriculture actually. Okay, that is why I told there are recombinant protein proposal, but into the market it is very less. Okay, in recombinant DNA technology, antibiotics are used. Why? Why we are using antibiotic in the media? Answer is there in the question only. Yes, antibiotic is used against bacteria to make bacteria free. Okay, answer is there in the question only. Example for a gene therapy is introduction of the gene for ADA for the people who are suffering from SCID. Okay, CID, right. Next, match the following now. You can do it very easily. You will do it now. Do now.
Option D.
Option D. No.
Option D.
Option D. See. BD cotton, baseless syringes. ADA, gene therapy. See, RNAi is a cellular defense mechanism. Got it? So it is your D. D. Next, first hormone prepared by genetic engineering. Insulin. Next, which of the following statement regarding the structure of pro-insulin and mature insulin are not correct? What is the point about pro-insulin and insulin are not correct? Read the statement correctly and then tell me which one. Which one? Okay, pro-insulin is made out of 3 polypeptide chain. Yes, ABC, true. C-peptide chain with the 33 amino acid are removed before formation. True. Mature insulin is made out of 51 amino acid. True. A and B together. Next, polypeptide chain A has got 30 amino acid and B has got no, that is wrong. Then polypeptide chain A and B are interconnected by only one. No, it is 2 disulfide. So it is your 4 and 5. So it is your option is C. Next, in gel electrophoresis, separated DNA fragments can be visualized by right. Next, then what is this acetylcarmine? Did you use acetylcarmine?
Yes.
Which technique to see what type of cell division? Mitosis. Okay, they have given this thing you would have used. It is your acetylcarmine only you use. Mitosis. Next. Then 49, through green revolution has been a resounding success in terms of agricultural production. Yet it has failed in its overall social objective. Because why? Tell. Green revolution was success initially, but they could not make up. Why? So actually when a green revolution started in, first thing is with the population increase, they want to increase the production also. So what they have done is like go in for use of fertilizers to a greater extent. That is the logic. Then fertilizers have started to cause a harmful effect. At the same time, the rate at which the population is growing is not able to manage the production also. Then they shift to rDNA technology. This is a story, right? Now read the option and tell the correct answer. All of these. All of these. Next. Given below are 4 statements pertaining to separation of DNA fragments using gel electrophoresis. Identify the incorrect statement. Now you can only do because half of the questions we have done, almost done. So now here after last time you should be able to tell right answers now. Tell now. Tell now. Do all of you accept option D? C. Then A, B is left out. No, that also you can. We'll mark. Okay, now DNA is negatively charged molecule, so it is loaded on gels. Yes, right. Towards the anode terminal. It is towards cathode terminal. So that is an incorrect statement, right? Because DNA is negatively charged, it will move towards anode. So that is incorrect. It will move towards the anode terminal, it will move towards the cathode terminal. Next, DNA fragments travel along the surface of the gel whose concentration does not affect the movement of the DNA. No, it will affect it. Based on, yes. So second is also wrong. Third one, smaller the size of DNA fragments, larger is the distance it travels through it. Right, farther it will move. Pure DNA can be visualized directly by exposing UV radiation. No, how do you see it? With titanium bromide you will observe it. So the option is C. Correct. Whoever has told D, it is right. If we ask next option, immediately one more option will come. So it is D. 1, 2 and 4 are incorrect statements. Okay. Next. 51st. Okay, or 51. Study the following statement and select the incorrect ones. Select now. You can. It's a very direct question. Tell me the answer now. Option C. Okay, fine. Then next, and I did not tell the answer. They have given. One more option will come anyway. 4 options are there. 1, 2, 3, 4. I get all 4 options right. Tell now. Read and tell. Okay, first one. Okay, fine. Whatever. Golden rice is a transgenic variety of Oryzae sativa. It is rich in beta carotene. Yes, just now I told her. It will produce vitamin A, but genus beta carotene. Got it now? So it is right. Next, flavor saver transgenic tomatoes remain fresh for a long time. Yes. Then the anticoagulant uridine is being produced through the transgenic Brassica napus seed. Yes. Actually, they took the gene from the leech. Got it? Because leech, when it sucks the blood won't coagulate when it is taking in. So it has a gene. Gene is taken from the leech and it has been introduced into a plant. Then if it is introduced into the plant, what is a vector? If it is introduced into the plant, what is a vector? Agrobacteria only. There is no second thought. Got it? So automatically the seeds of that plant started to produce. So they can take the seed and they can extract that product. You did it. Got it now? Then next, last option. So that is also right. Got it? Then next, BT. In BT cotton indicates that it is a genetically modified crop produced through biotechnology. Right or wrong? Wrong. Why wrong? Because all three I told right. You have to tell me the reason now. Read the question again. Answer is there in that. You know the answer. BT in BT cotton. What does BT in BT cotton refers to? But what they are giving, it is genetically modified crop produced. So it is wrong. So option is A. 1. You understood? Read the sentence twice for statement questions. Next. Following statements describe the characteristics of an enzyme restriction endonucleases. Identify the incorrect. Now you can do it. A. You only decide and tell me which is the right answer, because already we have seen A is the right answer, right? A is the right answer. Because DNA, the enzyme binds DNA only to a specific site of one content. So it is A. Repeated question. Okay, fine. Which of the following risks are associated with genetically modified food? Toxicity in humans, allergic, antibiotic resistance of microorganisms, all of this. All of this. Which of the following is a restriction endonucleases? Repeated at this also. End to. Which of the following statement is correct regarding the process of RNA interference? First time we are coming across. Now read and then tell me the answer. Correct one you have to find out. Okay, which of the following statements is correct? Okay, now RNA interference is used to prevent the infestation of protozoans. Correct. To prevent a protozoan infection. Correct. Okay, now hit C. She has already told C. Okay, next. But it has given infestation, not infection. Okay, next. Now option change. B. It takes in some eukaryotic and all prokaryotic organisms as a method of cellular defense. No, only in eukaryotes or prokaryotes. You will find this. Okay, in eukaryotes. Because plant only you are using, no. So not in both. So it's a eukaryote. So that is also your incorrect statement. The method involves silencing of a specific messenger RNA due to complementary DNA. True. Correct. Then this is a novel strategy to produce a true. So it is option A. Got it? It is option A. Now golden rice, you can only tell. Option B. Then 57, a monopoly which is granted to a person who has invented new and a useful article. What is that? Monopoly. Monopoly means to one person only you can give. Bioperson is a one-to-one. It's a granted to a person, individual, who has invented. Patenting. If I know the result, if I identify, I will patent. So individual it is. Now which one of the following is a correct statement? Now again, I think this is also repeated. Yes, but options are not same, I think so. Options are not same, right? And now you tell me. Which one of the following is a correct statement? C is the correct. Because here they are asking correct. Okay, now let us discuss why D is wrong. It's not ethylene. Gene is wrong. It is an adding a copy of polygalactourinase enzyme. Again, antisense technology only they will follow here also. Next, anticoagulant uridine is being produced from the transgenic brassica. True, right. Somatic hybridization involves fusion of two complete plant cells. That is right or wrong? Huh? Wrong. Why you are saying it wrong? What is wrong in that? It is crossing of two pure lines and then artificially selecting the progeny and then of decided trait. Here they have given that two complete plant cell carrying decide gene. No. First they will two pure lines, then if you get a decided gene in them, then you select. So it is wrong. Got it? Next. BT indicates genetically. Again, same thing he has given. It is wrong only. Next, which one of the following is regarded as a natural genetic engineer? He is doing it naturally. Got it? Next, because he himself can enter into and cause a disease. So if you have to just insert the gene. Okay, now which is the criteria for DNA fragment movement on agarose gel during gel electrophoresis? Read the option correctly. B, because so larger the fragment size, farther it moves? No. B is right. Positively charged fragments move. Positive and negative it doesn't relate. It is based on molecular weight, So it is your B. Understood? At least a overview you should be able to think and then